This is a GCMS instrument, a gas chromatography mass spectrometer. This type of instrument is heavily used in environmental analysis and forensics lab. GCMS is really two instruments coupled together. A cheaper gas chromatograph and a more expensive mass spectrometer. Your sample will travel inside the column installed inside the gas chromatograph in the gas phase. Then it will be ionized inside the ionization chamber of the mass spectrometer. The resulting ions will travel at high speed through a mass quadruple until they hit a detector. The gas chromatograph is basically an oven with an injection port connected to a tank of clean and dry helium gas. Inside the oven, a capillary column such as this one is installed and helium gas is flowing through it. The temperature of the oven and of the injector can be ramped to target temperatures at specified rates, thus allowing the sample to travel in the gas phase from the injection port through the column all the way until it exits the column and enter the mass spectrometer. The capillary column length is typically 30 or 60 meters long, and its diameter is usually less than a millimeter. The inside wall of the capillary column is coated with a phase, a few micrometer thick. This phase is made of silicon-based compounds that are either polar or nonpolar. Depending on these compound properties, the sample will be more or less retained inside the column as it travels through it. Thus, a mix of compounds will separate inside the column as they travel through it. Some compounds being retained for a longer time inside the column, while others will fly through it in just a few minutes. Upon exiting the capillary column, each compound, now separated from others in the mix, will be ionized in the high temperature, 210 degrees Celsius, ionization chamber by electron ionization. This chamber is under very high vacuum and magnetic field. The gas phase molecular ion will undergo fragmentation. Then it will be submitted to a rotating electrical field inside the quadrupoles chamber, which is 4 cylindrical rods parallel to each other. Opposing rods carry the same charge and are electrically connected. The charges vary from positive to negative. The resulting trajectory of the ions is a spiral at a distinct speed towards the detector. The mass spectrometer is routinely calibrated to determine which masses will hit the detector and when. Using this calibration curve, the software can calculate the mass of the unknown ion. You will need to prepare your sample in a very dilute solution. Concentration must be down to a few parts per million, because the mass spectrometer is a highly sensitive analytical technique. You will also need to prepare a vial of your wash solvent. Your sample will be in a smaller 1.5 millimeter vial, and you will position this vial on the sample tray. Up to 10 sample vials can be positioned on the sample tray using your autosample. You can thus program an experiment to measure up to 10 samples in a fully automated mode. There are 2 softwares controlling this instrument, GC Bruker and MSWS. Open the GC remote control software, then open the MSWS software. You will only enter your parameters and analyze your data in the MSWS software, but the GC software must be open in the background. Enter your GC parameters first. Click on the MS Editor button, then select 43X GC control. By selecting successfully autosampler, flow and pressure, injector, and oven, you can program the oven, the autosampler, and the injector. Start and end temperatures, heating rates, isothermal steps, number of washes before samples, and refer to red binder for details. Pay attention to the split condition. Split mode refers to the possibility of diverting a portion of your sample outside of the capillary column to prevent from overloading the column. Enter your mass spectrometer parameters. Select Scion mass spec control acquisition method to program at what time during the experiment the mass spectrometer should start analyzing ions, for how long the analysis should last, and the ions mass range. For details on this, refer to the red binder. Save your methods into the method folder, then create your sample list and select your data file by clicking on Acquisition button, then selecting Sample List and Data File. Then position all your sample vials and wash vials on the autosampler and hit Begin. The run will begin. You'll see that the gas chromatograph displays the temperature range and equilibrium status. Once the gas chromatograph is ready, the syringe installed on the autosampler will rotate and aspirate the wash solvent a few times to rinse. Then it will rotate and aspirate your sample a few times to rinse with your needle above the injection port. The number of times the syringe will aspirate the wash solvent and your sample can be programmed through the software parameters. The syringe will lower at once and puncture the septum inside the injection port and inject your sample, all within a fraction of a second. The sample will travel inside a tube placed between the injection port and the syringe called the injection liner. The temperature of the injection liner can be programmed through the software. At the end of the injection liner, your sample should be gas phase and will be carried by the flow of helium all the way to the end of the capillary column. Inside the capillary column, your mix of compounds will separate based on their interaction with the phase of the column. This is called the retention ability. One after another, they'll exit the column and enter the ionization chamber where they'll undergo ionization, then fragmentation. For data analysis, simply click on the Data Process button under Task pane and select your data file. The GC plot will be displayed. If you left-click the mouse with cursor on any peak of the GC plot, the resulting mass spec, the ion fragmentation spectrum will be displayed below. You have the options to calculate the area under the curve for quantitative analysis. For details on how to do this and more, please refer to the red binder.